Hello, I'm Iwona Wójcik. I'm from Poland, and here in Genos I'm doing my PhD studies. I'm enrolled in industrial PhD. Genos, based in beautiful Croatia, is an industrial leader in high-throughput glycomics and glycoanalytical technologies. And we as a team are fascinated by glycans. Glycans, complex sugar molecules, are everywhere. They are on the membrane of every living cell. They can be attached to proteins and lipids, modifying their properties. More, they are changing in many diseases and in response to environmental factors, such as our lifestyle or environment. It means that actually they can tell us something about the status of our body. So signs are there, but first we need to learn how to read them. And here we are. Our aim to have a closer look at glycans. Equipped with the state-of-art instruments and automated workflows, we characterize glycan monosaccharide composition, structure, position on the protein, and measure their abundance. Finally, we would like to find glycan-related changes in biological fluids of patients in many pathological states. We are fascinated by glycans, but even more fascinated by developing high-throughput technologies and analytical strategies that are required to measure all those glycans. Currently, we are collaborating with Waters, and we had the pleasure to test the newest BioAccord system and GlycoWorks RapidFluor kit. Both allowed us to analyze glycans more precise than ever before. Do you know how this solution helped us to be so successful in the glycan analysis? Let me explain. We are analyzing glycans and glycopeptides from immunoglobulins isolated from the human blood. Sounds difficult? Not with BioAccord. The system comes with ready-to-use optimized workflows for released glycans and glycopeptide mapping analysis, so we could start running our samples right away. For more complex samples, the system can be easily optimized, like we did in case of glycans released from the human plasma. The only one thing which is left for us is the final check and interpretation of the data. The system combines leading UPLC technology with a high-resolution mass spectrometer within one solution. This combination allows us to analyze complex biological samples using two different analytical technologies. Now we can directly integrate fluorescence and MS signal, which enables us to characterize glycosylation changes more precisely. Now we can see even more. We are experts in mass spectrometry. We used to set up, maintain and run instruments in the lab. We know that LC-MS systems are complex and not necessarily easy to use. But the BioAccord, which is a robust and easy-to-operate platform, completely changed our point of view. Setting up an instrument is nothing more like a simple one-click operation. After that, the system auto-tunes, calibrates and diagnoses itself. This process takes just a couple of minutes, and the result we will see in the front panel of the detector, which indicates if the system is ready or gives an alert if anything needs to be checked. Any issues are rapidly diagnosed and resolved. In short, there is no need to be a glycan and mass spectrometry expert to obtain high-quality data. This is an instrument that you can buy, plug in, put your glycan samples in and start your measurement. And we have more time to focus on more meaningful questions. Do you wonder if BioAccord can cope with hundreds of biological samples to be analyzed? The answer is yes. As a high-throughput glycan laboratory, we need something that is precise, robust and accurate. The BioAccord passed the exam and ensured us with high quality and consistent data. From our experience, we can say that the BioAccord allows comprehensive analysis of glycosylation. At the same time, the system made our analysis easier, more meaningful and effective. Having this smart technology on board, we advanced our analytical methods, and this helped us answer critical biological questions more effectively.